Lugano, we're also presenting work on our genomic analysis of tumor progression in CNS lymphoma to test the hypothesis that genomic aberrations at the setting of progression or at final progression may be different from the initial aberrations that are associated with the onset of disease. And one of the goals of this is to identify mechanisms of resistance to therapy. And so we've done this in a collaboration with investigators at Stanford to identify in autopsies mutations that are distinct from diagnostic specimens. And this is a multi-regional analysis in which we take specimens in autopsy brains that contain lymphoma, sequence them for genomic aberrations compared to normal brain to look for lymphoma-specific aberrations and map them to the trajectory of lymphoma progression anatomically. And we've shown that indeed lymphoma, at each step in lymphoma progression, there's evidence for new mutations as well as some evidence for new copy number aberrations which are anatomically distinct in some cases in the brain, which is perhaps a novel finding. And some of these aberrations may be associated with increased fitness of the tumor and its ability to withstand multiple therapies. So many of these patients received not only methotrexate, but went on to receive radiation as well as IMiDs such as Lenalidomide and Pomalidomide. And that's perhaps one of the more interesting findings in this small study is that the tumors that were associated with Lenalidomide or Pomalidomide exposure have novel mutations in the Ikaros pathway, and that may be a mechanism a novel mechanism by which the tumors become resistant to IMiDs. We also see downregulation of cereblon expression in tumors, which is an established mechanism of resistance. We see downregulation of cytotoxic T cells in diagnostics in autopsy specimens, significantly compared to an independent set of 20 diagnostic specimens, suggesting that there are multiple immunosuppressive mechanisms which contribute to resistance at the time of final progression as well.